In microbes, we are studying about viruses and bacteria. So already we have studied what are viruses, types of viruses, and the replication in virus, and the economic importance of virus we have studied, and also the classification of viruses based on nucleic acids and based on host. Then next, in the bacteria, we are studying again about the structure of bacteria, types of bacteria, and reproduction in bacteria. In reproduction, we have studied about the asexual reproduction, that asexual reproduction takes place by fission, binary fission, and as well as endospore formation. So during favorable conditions, when the bacteria are living in a favorable environment, the bacterial cells, they divide for every 20 minutes. So in a period of 20 minutes, the existing number of bacteria double in number. So a single bacteria become 2 in 20 minutes. In the next 20 minutes, the 2 becomes 4 bacteria. Then in the next 20 minutes, 4, 8. Like that, the number goes on double for every 20 minutes. That is by binary fission. But however, when there are unfavorable conditions, when there are certain abnormal conditions which does not favor for the growth and development of the cells, there the cells they are about to get destroyed or they die. In such circumstances, some of the bacterial cells, they divide, they do not divide. That means they form a thick cell wall, and the cell protoplast remains within the thick wall, and this is called as endospore. So this endospore can withstand very dry conditions and extreme temperature conditions, and then on the oncoming favorable conditions, when the favorable conditions arrive, there is the removal of the protoplast from the thick cell wall, and thus the bacterial cell continues to divide by binary fission. Now in sexual reproduction, where we call it as usually bacterial recombination. So that bacterial recombination is a type of sexual method, but strictly when we say sexual method, there is no male and female in case of bacteria. But however, one of them is considered as male and the other as female. Now this is referred as the bacterial recombination. Now this bacterial recombination, it is characterized by the DNA transfer from one cell, one bacterial cell, to another bacterial cell. So the one cell which gives the DNA, it is called as the donor. The cell which receives DNA, it is called as the recipient. Now in genetic recombination, there is a transfer of DNA from one cell, which we call it as a donor, to another cell which it receives DNA, it is called as a recipient. So this transfer of DNA takes place by 3 ways. That means there are 3 methods of bacterial recombination. So one such method is conjugation. So conjugation is one method. Then there is the transformation, which is a method of genetic recombination, and there is transduction. This is another method of genetic recombination. So conjugation, transformation, and transduction. These are the 3 ways of bacterial recombination, where DNA is transferred from one cell to another cell. This is the main theme of this process. Now during conjugation, what takes place? So during conjugation, there are cells of different strains. Suppose these are the cells, bacterial cells. Now this takes place between a F plus strain and HFR strain. So conjugation takes place between F plus and F cell, HFR strain and the F strain. So these are the strains of bacteria. So the DNA from this, suppose this is the DNA, and here this is the DNA. In this, the DNA of the F plus or the HFR strain. So F means it is a factor. It is a type of DNA which is present in the bacteria. HFR means actually it is high frequency. So this high frequency DNA which is present in this is transferred to the cell which does not have that DNA. Now this takes place between two cells. Actually this method was described by two scientists called as Ullmann and Jacob. So Ullmann and Jacob in 1956 they discovered conjugation in bacteria. Now what this process or how this process takes place? Now this, in this there is a contact between two bacteria of F strain, or it is also called as F minus strain, and F plus or HFR strain. So when this HFR strain contains, so F plus strain is one type of bacteria, HFR strain is another type of bacteria. The strain, it may be in any species. This is F bacteria, F strain or F minus strain. That means there is no this type of DNA in this. Now these two cells, they lie side by side. They are close to each other. Now in the F plus and F minus strain, difference is in the F plus and HFR strain, there is a specific type of flagella which is called as sex pilus. So it is a finger-like projection actually. It is not a true flagellum. A flagellum-like structure it is. It is a long tube-like which is present on the F plus and HFR strains. And these sex pilus is not present in the F strain. So this sex pilus comes in contact with this bacteria. So when the bacteria enters into that, or when the two cells come in contact, the DNA enters through this sex pilus and gets transferred into the F cell, F minus cell. Now the DNA means it is a part of the genome, some DNA fragment. Now in F plus strain, what happens? Only a small, that means suppose when a F plus and F gets in contact, when a HFR strain and F comes in contact, when there is F plus in contact with this, there is a part of the genome, a small part of the genome is transferred to this F minus. So this a small portion of the genome, in other words a small fragment of the DNA is transferred from F plus to F. So if F plus and F are in contact with each other, a small part of the DNA, a small part of the genome is transferred to the F cell. Suppose if HFR, high frequency strain, is in contact with this F plus, a large part of the genome, a large portion of the DNA is transferred to the F cell. So that is the difference between F plus and HFR strains. So F plus will donate only a small part of the genome, and HFR, high frequency strain, donates a large amount of genome, a large part of DNA to the F cells. Now these F plus and HFR genomes, they are actually called as episomes. Episomes, they are some part of DNA, some fraction of fragment of DNA present in that cell. So these episomes are present in the F plus and HFR strains, and during the pairing of this two cells, what happens? The DNA is transferred, means when the two cells come in contact with the sex pilus of the F plus and HFR, sex pilus is absent in F strain. Sex pilus is present only in F plus and HFR strains. So that the DNA or the genome is transferred through the sex pilus into the F strain. Now the result is what happens. So what is there in the transfer of DNA from one cell to another cell? So when the DNA is transferred from one cell, that recipient cell gets new characters, and this F minus strain undergoes genetic recombination, means it has additional genes, it has additional characters. So it gets additional DNA from the donor cell, and that is how the F minus strain gets that genome fragment, small fragment in case of F plus, large fragment in case of HFR. So there is recombination taking place between the donor fragment and the recipient DNA. So the recipient DNA will receive the donor fragment, and thus results in genetic recombination. Now this this is what is called as conjugation. So conjugation results in recombinants. The recipient cell which gets foreign DNA and acquires new characters is called as a recombinant. So this recombinant can be done, or this conjugation can be done to produce recombinants when the cells are placed in a suspension of F minus and HFR cells. So the HFR cells and F minus cells, so they cause conjugation, and these recombinants which are produced, which are formed, they are variable in their genotypic constitution and also in the phenotypic expression. So in the genotypic also they are changed, means they vary. So that is genotypic constitution. So phenotypically also they will change because of new DNA. They will express a different character from the former characters. So that is about the conjugation in bacteria which takes place between such kind of strains. Next, about transformation. So transformation is a process which is also the transfer of DNA from one bacteria to another bacteria. But here the transformation is usually done between dead cells and living cells. So this aspect we have already studied in the second PU by knowing the Griffiths experiment in genetics. So Griffith experiment, what actually Griffith did. So Griffiths experiment means Griffith actually studied this transformation in the year 1928. Later Griffiths experiment was demonstrated by three scientists called as Avery, MacLeod and McCarthy. So they demonstrated this experimentally. In other words, they continued Griffith's experiment. Now what actually this transformation means? Frederick Griffith, he used a bacteria which is called as Diplococcus pneumoniae. He took a bacteria called as Diplococcus pneumoniae, also referred as Streptococcus pneumoniae. So this Diplococcus pneumoniae it is having two strains called as smooth strain and rough strain. So this S strain or smooth strain and rough strain differs in the cell wall. This smooth strain is actually smooth in appearance and it is having an additional cover on this cell called as the capsule. This is capsule. Here there is no capsule. So this capsule is present in S strain, smooth strain. In R strain it is absent. In other words, it is capsulated, non-capsulated. So it is smooth. Because of the capsule, the cells appear smooth in appearance. They appear smooth. Here there is only cell wall. The cell wall is rough in appearance. Hence it is called as a rough strain, R strain. And also in another important thing is S strain is virulent and this is non-virulent. This is also another important. That means if S strain is present in a person or an individual, it causes pneumonia. Diplococcus pneumoniae causes pneumonia. S strain causes pneumonia, but R strain does not cause pneumonia. So S strain is virulent. It causes pneumonia. Suppose when we introduce S strain into an individual, into an animal, it causes pneumonia. Suppose if you introduce R strain, it does not cause pneumonia. That is the difference between S strain and R strain. So this now Griffith, what he did is he injected S strain, means living S strain into mouse. He injected and the mouse died of pneumonia. Means the mouse was infected with pneumonia, so they died. So naturally, so already we said that S strain is a smooth, capsulated, virulent. It causes pneumonia. Rough strain is rough, non-capsulated, non-virulent. So it is non-capsulated and non-virulent. So it does not cause pneumonia. So naturally, when you inject S strain into mouse, it causes pneumonia. So the mouse died of pneumonia. Similarly, if you introduce R strain into mouse, the mouse not die. They do not die. Definitely. They do not die. And next what he did is, he took heat killed S cells and injected into mouse. Here living R cells, when they were injected, so S cells died, caused the death of mouse. R cells did not cause death because they do not induce pneumonia. They do not cause pneumonia. However, when heat killed S cells were injected, so the mouse not die naturally, because S cells are not living. Living S cells, if you inject, they die automatically. But when the same S cells were heat killed, means when the S cells were subjected to steam and they made inactive, so they cannot cause pneumonia. That is why the mouse did not die. They did not suffer from pneumonia. This is what happens next. In the next step, what he did is, he took heat killed S cells combined with the living R cells. These two were injected together. See, heat killed S cells only do not cause pneumonia, no death. When the heat killed S cells combined with the living cells injected into mouse, so the mouse died of pneumonia. So though the heat killed S cells alone cannot cause pneumonia, but along with the living R cells, they caused pneumonia, or they together caused pneumonia. Now by this is what actually Griffith did. Now by this, what Griffith came to know is, there is some transformation principle, there is some substance which acts as a transforming principle present in S cells. So even when the S cells are died, when they are heat killed, so that transforming principle or that substance has entered into living R cells and caused pneumonia. Living S cells caused pneumonia, heat killed S cells no pneumonia, but heat killed S cells with the living R cells caused pneumonia. So Griffith concluded that there is some transforming principle and there is some substance which entered into the living R cells and caused pneumonia. But what is that substance? He did not explain. What nature of that substance, whether it is a carbohydrate, whether it is a protein, whether it is a lipid fat, or whether it is any other nucleic acid, or whether it is any other substance. So Griffith did not mention the nature of the transforming principle, but later three scientists, Avery, MacLeod and McCarthy, after the Griffiths experiment, they continued Griffiths experiment. So they did not did a new thing, but whatever Griffith had done, they continued the same. Now what they did is, they predicted that the transferring principle is DNA. So they extracted the DNA of the heat killed S cells. So the DNA extract was taken from this heat killed S cells and mixed with various enzymes. So the DNA extract taken from heat killed S cells was mixed with the amylase, was mixed with the protease, was mixed with RNase, was mixed with the DNase. So when the DNA extract from S cells, so they did different step. The DNA extract was mixed with the amylase first and injected into mice. When it was injected into mice, so the mice died of pneumonia. That means amylase did not destroy the DNA in the DNA extract, and that is why the DNA caused the pneumonia. Similarly, the DNA extract was mixed with the protease. So there also the protease only acts on proteins. The amylase acts only on carbohydrates. So the DNA caused pneumonia and the mice died of pneumonia. Similarly, when the DNA extract was mixed with RNase enzyme, which destroys RNA, so there it was injected. So there also the mouse died of pneumonia, because RNase does not destroy DNA. Then in the next step, the DNA extract was mixed with the DNase enzyme and injected into mouse. So there the mouse did not die, because DNase enzyme destroyed DNA. So this DNase enzyme destroyed DNA, and when the DNA was destroyed, the DNA could not express pneumonia, and hence the mouse did not die of pneumonia. That is how Avery, MacLeod and McCarthy, they showed that the transforming principle, the substance which has entered from heat killed S cells to living R cells is DNA. So that is why the R cells have been transformed into S cells by the DNA of the S cells. So the R cells have acquired new DNA. The living R cells has received the DNA of the S cells and acquired new characters, means there was a genetic recombination. They have been transformed from R strain to S strain. So they have become now S strain because they cause pneumonia. This is what actually Griffith conducted the experiment on Diplococcus pneumonia, and it was continued by Avery, MacLeod and McCarthy. Then if you come to transduction, now in transduction, here also the same thing. Now suppose this is a bacterial cell. So when a bacteriophage, suppose this is a bacteriophage. So the DNA enters into this. The DNA of the bacteriophage. Now this DNA of the phage, that is the phage DNA. So it was actually discovered by Lederberg and Ginder. So Lederberg and Ginder, they have expressed this method of genetic recombination in bacteria. Now Lederberg and Ginder, what they explained about transduction. It usually occurs in bacteriophages. That means when a bacteriophage infects bacteria. Now especially he observed this in a bacteria which is called as Salmonella typhimurium. So in this bacteria they observed this type of genetic recombination. So transduction means it is also transfer of genetic material, DNA, from the donor to the recipient. All the three are same. DNA is transferred from donor to the recipient, but here it is transferred through a phage, through a bacteriophage, phage particle. So when a phage infects a bacteria, so what happens? When a phage infects a bacteria, the DNA of the bacterial cell, so it breaks down into small fragments. So this breaks down into small fragments like this. Now this host DNA, that means the bacterial DNA, when it breaks down into small fragments, it undergoes replication along with the viral DNA. The viral DNA also gets multiplied, and some of the DNA fragments, they are incorporated with the virus particles, becoming a transduction one. So some of the DNA gets incorporated. So this DNA which gets incorporated, it gets a new character. So when it remains in a particular cell, it becomes a transducing one. With the existing DNA, new DNA is joined from virus. So the bacteriophage is the responsible for the transfer of DNA. So it is the transfer of DNA which is mediated by the phage or by the bacterial virus. That is how Lederberg and Ginder, they explained that the bacteria which are infected with these particles, phage particles, their transduction takes place and the DNA of the virus particles undergo genetic recombinations with the bacterial DNA. So that phage DNA or the DNA of the virus, it will undergo recombination with the DNA of the bacterial cell and thus acquire new characters. So those are the three genetic recombination factors, or the three genetic recombination which takes place. Then if you come to the economic importance of bacteria. So bacteria are both beneficial and harmful. So in general we can say, but bacteria has most benefits, but there are some harmful effects, like bacteria cause diseases in plants and animals. Now one by one, bacteria helps in nitrogen fixation, that we have studied in nitrogen cycle. So bacteria, they help free living soil bacteria, they fix atmospheric nitrogen and they increase the fertility of the soil. And there are bacteria which are present in the root nodules, which are seen in the leguminous plants, like rhizobium bacteria. So that rhizobium bacteria helps in the fixation of nitrogen. So free living soil bacteria like Clostridium and other bacteria, they help in the fixation of nitrogen. And bacteria also are used in industry. Various industrial products are obtained from bacteria. The common example we know, Lactobacillus used to convert the milk into curds, and also in the production of butter. And this bacteria is characteristic in giving a taste and flavor in making cheese also. That is the use of this bacteria. And there are bacteria which are used in the conversion of sugar into vinegar. For example, Bacillus aceti is one bacteria which is used in converting the sugar into vinegar. And similarly, butyl alcohol and acetone, they are also manufactured from molasses, an intermediate product in sugarcane in sugar industry. So there the industrial products are used, are prepared. And also this curing of tobacco leaves, drying of the ripening of the tobacco leaves, dry leaves, is also done by bacteria before it is used as tobacco for chewing. And then curing of the tea is also done by certain bacteria. That is in case of industry where bacteria are used. And the hides and skin of certain animals, after drying, it is steeped in certain fluids containing bacteria, and it is used in the tanning of leather. So tanning of leather industry, there the bacteria are also used. And retting of fibers, removing fibers from the plants. Fibers are present in leaves and stem. So by the action of certain bacteria, where fibers get separated, we call it as retting of fibers. So the soft tissues present in between fibers, they are decayed, and the fibers are easily separated by the action of bacteria, especially in plants like this flax, hemp, jute, and even coconut also. Then putrefying bacteria, where the bacteria help in the sewage under anaerobic conditions. So the sewage treatment, the solid part of the sewage, the solid contents and the semi-solid contents of the sewage, they are decayed and liquefied by bacteria. And this liquid, after drained, it can be drained to the river, and also it can be used in irrigation fields. That is the use of putrefying bacteria. Then ensilage means it is the preservation of green fodder in pits by certain bacteria. So where the green fodder is preserved by a particular process using bacteria in pits, that is called as ensilage. Then antibiotics are prepared from bacteria like streptomyces, streptomycin. Then the chloromycetin antibiotic is obtained. Like this, there are various kinds of bacteria which belong to a group called as actinomycetes bacteria. Then in genetic engineering, biotechnology, production of human insulin and growth hormone, bacteria are used. So bacteria are used in the production of human insulin called as humulin, and also this growth hormone, where the bacteria are used in this production of this products, which we call it as gene manipulation or genetic manipulation. Then in the pest control, Bt cotton, we know that Bacillus thuringiensis bacteria is used to control a particular species of pest, where it produces a toxin called as Bt toxin. So there this Bacillus thuringiensis is used, which is a specific insecticide, and it is eco-friendly. That means it is not bringing about pollution. Then bioremediation, by removing the pollutants in contaminated water and soils. Certain pollutants can be removed by the action of bacteria in contaminated water and soil. Especially in oil polluted water, hydrocarbons can be neutralized by some bacteria, and thus water can be used for various purposes, for cleaning and also irrigation. Then in case of digestion, certain bacteria like ruminococcus, they digest cellulose in case of ruminant animals. So in ruminant animals, ruminococcus bacteria is present. It helps in the digestion of the cellulose by secreting the enzyme, cellulase enzyme. So that is in case of this ruminant animals. So these are some useful or beneficial aspects of bacteria. Now if you come to the harmful effects, bacteria causes diseases in plants and animals. And in plants we can mention some few diseases like citrus canker and ring rot of potato, then crown gall of apple, and many more. So these are some plant diseases. In human beings, there are many diseases like cholera, typhoid, pneumonia, just we have mentioned here, and tetanus, tuberculosis, dysentery, plague, whooping cough, influenza, diphtheria, and etc., like syphilis and others. And even here, bacteria cause loss of fertility. That means denitrifying bacteria, the soil which is containing nitrate, it is converted into nitrite, where the fertility of the soil is lost. So that is loss of the fertility in case of denitrifying bacteria. Then food is spoilt by certain bacteria when they grow on foodstuffs and they spoil. When foodstuffs are unprotected, that food is spoilt, and here they release a kind of gas. So when such spoilt foods are taken, when they are consumed, it results in a state or a condition called as botulism. So that is one harmful effect of bacteria. So bacteria, especially they produce plant diseases and animal diseases, loss of fertility and food spoilage. So these are the harmful effects of bacteria.